So what I'm going to present to you today is more of a cell biologist's view on Borrelia, and especially on the interaction of Borrelia with primary human macrophages. So I don't have to remind this audience that Borrelia, and especially Borrelia burgdorferi, is an important pathogen. I want to stress the fact that while we have been mainly using Borrelia burgdorferi, we also have used Borrelia afzilii and also other species. So the mechanisms of uptake and intracellular processing I'm presenting to you are fairly concerned in terms of species. So I'm going to refer to that mostly as a Borrelia species in a more broader term. So one of the things I want to draw your attention to, and all of you know that, is one of the earliest signs of an infection with Borrelia can be erythema migrans, the skin rash. And this skin rash is especially enriched in human immune cells. So this is the first site where the human body is actually battling an infecting spirochete. And one of these immune cells are human macrophages, and this is what my lab is focusing on: how are human macrophages interacting with infecting spirochetes. One thing you should remember, all of you know that Borrelia is a spirochete, so it has a helical shape. This will be important in one of the sections of this talk. And I also want to remind you that Borrelia contain periplasmic flagella, and these are organelles that enable very fast movement of these spirochetes, and this is also important for the interaction with the macrophages. So what my lab is actually focusing on is how Borrelia is being taken up by the macrophages, and once it's been taken up, how it interacts with the intracellular machinery for degradation. So, and these will be the two main foci of my talk, and there will also be a small bonus, let's say, of unpublished stuff later in the talk. So first of all, what does it look like? As I said, I'm a cell biologist by training, so we're like to looking at cells. So I'm a great believer in actually looking at things and then trying to figure out what's the molecular mechanism behind this. So what you see here in red is a primary human macrophage. And this macrophage expresses a probe for actin filaments. So actin is one of the main constituents of the cytoskeleton of these cells, and the dynamics of the actin cytoskeleton enable lots of the morphology and the shape changes, and also the uptake protrusions that are involved in wrapping and internalizing the Borrelia. And in green, you see GFP expressing Borrelia, so they express green fluorescent protein, so they're naturally or artificially green. And if I start this movie, you see here, these flittering shapes here around are Borrelia that are not being captured by macrophages, that are still living happily in the petri dish. However, here you see two Borrelia that are already being captured and partially internalized by a primary human macrophage. I'm starting this movie again once it's finished. And what you can see here is that they're being enwrapped in actin-rich protrusions that really wrap around the helical shape of the spirochete. And this is what what has classically been referred to as a coiling pseudopod. And you see that there's actually lots of dynamics going on. So the F-actin the filamentous actin is constantly being newly formed and also degraded. So F-actin dynamics are really important for the uptake and internalization of Borrelia. So just a short excursion into F-actin dynamics. You may know that actin is present in forms of monomer or globular actin, but it can also form these long elongated filaments, and this is referred to as F-actin. And the transition occurs by polymerization. So basically several globular actin monomers nucleate together, and then spontaneously you have a polymerization of this actin filament. The critical state here is this trimer state, because this is energetically unfavorable. And the cell has devised methods to really overcome this energy barrier, and by that actually to locally regulate actin dynamics by the so-called nucleation factors. And this is also something that we're looking at in the lab. You may know, or you may have heard, that there are a couple of nucleation factors. One of the most important ones is the so-called ARP2/3 complex. And this complex here binds to the side of existing actin filaments, and then it leads to formation of a new daughter branched filament. You may not have heard that there are also other nucleators called formins that lead to the formation of unbranched, so just longitudinal actin filaments. And while ARP2/3 complex has a very defined and limited set of activities, basically just nucleates and leads to these branched actin networks, formins, there's a family of 15 or more formins in the human body. They have a set of activities so that they not only nucleate, they only elongate these filaments, they can bundle these filaments, they can also sever these filaments. So there's a lot more dynamic in the actin filament formation and turnover here. And mostly this leads to the formation of these straight longitudinal bundles of actin. So that just as a biochemical background. So, and these nice young ladies looked at the initial stages of the uptake of Borrelia by primary human macrophages. This is Xenia Nai, a former graduate student in the lab, and Ann-Kathrin Hoffmann, a former master student in the lab. What Xenia and Ann-Kathrin looked at were unstimulated primary human macrophages. So this is a three-dimensional reconstruction of the actin cytoskeleton of such a quiescent, unsuspecting, I must say, macrophage sitting in a petri dish, sitting here and adhering on the ventral surface. And here you have what we call the dome, which contains most of the nucleus and the cytoplasm. So this is actually quite a flat cell with an extension here in the C direction. And once an evil researcher adds Borrelia to the petri dish, this cell gets a shock and it responds. And what it responds with are these longitudinal protrusions called phallopodia. So these are actually sensors of the cell that enable the cell to detect in a very large, in an actual larger volume of space than the actual cell body, whether there's something threatening or interesting to eat in the near vicinity. And what Ann-Kathrin could show is that actually I might show that here, yeah, that the number of phallopodia rises dramatically once Borrelia are present in the petri dish. And Ann-Kathrin then also looked at all these different formins that I mentioned, and she identified 3 formins, FMNL1, MDIR1, and DEM1, and downregulated their expression in primary human macrophages by siRNA, and showed that by targeting each of these individually, you get a decrease in the formation of these phallopodia. So they're really important for the formation of these phallopodia. And here I'm going to show you for one of them actually how they're actually located. So in green, you see again the F-actin cytoskeleton of a primary human macrophage. Actually, that's just a part of the surface of this cell, so it actually extends to a much larger degree to the sides here. And these longitudinal extensions are the phallopodia. The Borrelia, you see here in this whitish color, they're already being captured by these phallopodia. And one of these formins, MDIR1, is in red, and you see that this is here very much at the tips of these phallopodia. This is a 3D reconstruction, and you see that the Borrelia are really encaged by these phallopodia actually. So, and if you play with the fluorescence intensity, you see that the MDIR really sits at the top of these filaments. So it's really is probably one of those formins that actually sits on the top of actin filaments where it drives also the nucleation. A similar theme emerges for FMNL1. However, it's differentially distributed compared to MDIR1. So here again, you see a primary human macrophage. Here you have an expression of FASN. FASN is one of the actin bundling proteins in the cells, just to highlight the phallopodia here in red. And here you see FMNL1 one of the isoforms called 1 beta. And this is a cell that has been stimulated by Borrelia. And you see that while the red Phallopodia is growing out, the FMNL1 comes with it. And this is better seen here in the gallery. So in contrast to this tip-like protrusion, tip-like localization of MDIR1, FMNL1 is actually localized throughout the whole body of the Phallopodium, which very nicely also coincides with its biochemical activities, which is not actin nucleation, but actually actin bundling. So it really results in a very much stiffer Phallopodium that is able to extend and protrude very directionally. And here again, the same for MDIR1, just to illustrate the fact that you see here very different localization here at the tips of these Phallopodia, and just also showing that these Phallopodia are actually very rigid structures that really do not bend, but they rather move as a complete structure to the side. Again, here seen in the gallery image. So a first step in the uptake of Borrelia is actually the capturing by these rigid elongated Phallopodia. What I've shown you and what has been known in the literature for decades is that one of the main and the most striking uptake structures is actually the coiling pseudopod. So, and what these nice little ladies have shown actually here is that this Phallopodia stage precedes the actual formation of the coiling pseudopod and then the major internalization step into the primary human macrophage. So this is here illustrating the fact that you have here this effect-enriched coiling pseudopod following the helical morphology of the spirochete and then really enwrapping that in alternating coils. And what Ann-Kathrin also could show very nicely is that one of these foramins, the third one, that is important for these Phallopodia formation is also important for the formation of the coiling pseudopod. So what I'm going to show you here is in blue, here you see the Borrelia going in and out of focus, and in green you see the DAM1, and this is the surface of the macrophage that it's in contact with Borrelia. Here you see the captured Borrelia. You don't see a Phallopodium, but here you see actually the DAM1 coming up, arising from the surface of the macrophage and actually following this helical shape of the Borrelia. And actually, if you knock down DAM1 by siRNA, you also get less coiling pseudopods. So DAM1 is one of the main players in the formation of these actin-rich structures. So this would be a first short summary. What we've shown is that Borrelia uptake is actually a two-step process. First, the Borrelia are captured and immobilized on the macrophage surface by these Phallopodia. And this is probably because the Borrelia are highly motile. So to enable the formation of this complicated coiling pseudopod, first of all, they have to be immobilized and kept near the surface of the macrophage. And this is done by the Phallopodia. And three of the regulators are these three foramins, MDIR1, FMNL1, and DAM1. And in the second stage, this immobilized spirochete can then be nicely and slowly be enwrapped by the typical coiling pseudopod that follows the helical shape of the spirochete. And here also DAM1 plays a role, and we've been showing way earlier that also ARCT3 complex plays a role. All of this is nicely published and actually quite old. More interestingly, the recent focus of the lab is more on the intracellular processing of the Borrelia. So what actually happens once Borrelia has been internalized in the macrophage, and how is it being degraded? And here also Xenia was instrumental in setting this up. And for a first assessment, we actually decided to look at wrapped GDPases, because if this is a cartoon of the cell, you see that you have various compartments that are involved in trafficking and taking up stuff and also in secreting stuff. And all of these mini organelles are positive for various proteins of the wrapped GDPase family. So these GDPases are actually important to drive the specificity and the identity of a compartment in the cell. So what Xenia decided to do is to look at internalized Borrelia that are being present in the form of membranous phagosomes, and then to look which rhabditidases are actually enriched at these phagosomes. And what you can see here is that especially two of these rhabditidases, RAB5A and RAB22A, are positive at these internalized Borrelia. So you see here this nice white coat of the RAB22A in a Borrelia that has been internalized by the macrophage. EA1 is actually an effector protein that works downstream of both of them. So this is very much in agreement of these findings. And this is actually what it looks like. So this is EA1. As I said, this is a downstream effector that is present at endosomes and is also being enriched at phagosomes that are formed at the uptake of specific particles. And what you're going to see here is that the Borrelia in green is being immobilized at the cell surface. Since no actin is stained, you can't see these uptake structures like the coiling pseudopod. But what you're going to see here is that the compartment that they are being taken up into is positive for EEA1. So here is the Borrelia. It's in contact with the surface, and then you see that it's very much sucked into the cell and being compacted in this ball of a phagosome that initially also is positive for EA1. So this elongated spirochete really becomes compacted into this tiny little ball, the step that we now actually refer to as a compaction event. So one of the things that interests us really is how this elongated form of the spirochete is actually being driven to this compacted stage, because we've observed that compaction is an absolute prerequisite for the subsequent degradation. So if no compaction happens, and for example, the Borrelia lose their phagosomal coat, they're also not degraded internally. So what are the mechanisms that drive actually the compaction of the Borrelia phagosomes? And just to illustrate this point again, here you see the elongated spirochete. It's being taken up in this phagosome that's positive for EA1, and this is also positive for RAB5. And also you can see nicely here that this is positive for RAB22A, and it gets progressively compacted. But, for example, as you can see here, that signal for RAB22A is much more intense than the one actually for the RAB5. And so one of the first things we wanted to figure out is what is the actual relationship between RAB5 and RAB22 in the uptake of Borrelia into phagosomes. And what we did here is we expressed both proteins in different colors. So the RAB22A you will see here in white, the RAB5 you will see in magenta, and the Borrelia as it expressing GFP you're going to see in green. Here you see that the Borrelia is already in contact with the macrophage. The macrophage has already started to internalize the Borrelia, and you see that RAB22A delineates the whole of the phagosome. And this here is still a part that is extracellular, and you see that the Borrelia is still wriggling around. So this is really driven by these periplasmic flagella, but eventually the whole of the spirochete is being taken up. It's internalized, it's compacted, and it's actually driven towards the nucleus, where also the lysosomes are that are necessary for the degradation. I'm going to play this movie again. And what you see here is that the earliest signal that we get is the RAB22A that really resides at the phagosome. And the RAB5A actually is not really present in the phagosomal coat, but is present at these little dots that actually represent vesicles. And these vesicles repeatedly contact the phagosomal coat. And this interaction is actually very critical for the compaction process, as we learned later on. Because this is another of these events, again, RAB22 in white, RAB5 in magenta. And what you can see here is that membrane tubules are being extruded and are shooting out from the phagosome. And this is actually what we think happens when the Borrelia phagosome is being compacted. You must imagine that there's a large surface of this initially elongated phagosome. And if you get membrane tubules extruded from this surface, you actually shrink the surface, and thereby you also shrink the phagosome, where it becomes more and more compacted, and then until basically you have a perfect sphere. And the site of this membrane extrusion events is always when you have a contact between RAP5A positive vesicles and the RAP22A positive coat. And what we think is what actually happens is that these vesicles give the starting signal for these membrane extrusion events. So no contact with RAP5A vesicles, no membrane extrusion, no compaction. This is actually the hypothesis that we've been working on. So you get a gradual compaction from an elongated phagosome into this sphere-like globular compacted form by successive extrusion of membrane material and progressive shrinking of the Borrelia containing phagosome. So actually that is why membrane tubulation events really captured our attention. And what Xenia could also nicely show is that these compaction events are very much decreased when, for example, delete or by siRNA the RAP5A in the cells. The white is the control. These are 2 individual siRNAs in shades of gray. They're actually just 2 shades of gray, not 50. So in RAP22A, you also get a very nice decrease. And actually the interesting thing is if you delete both proteins at the same time, you don't get an additive effect. So this means that both proteins actually work in the same pathway. And if you interfere at one point of this pathway, this is actually enough to disturb the machinery there. So RAP5A and RAP22A somehow work together in these membrane tubulation events that lead to compaction and eventual degradation of the Borrelia in these phagosomes. And to show that the compaction is actually necessary for the degradation, we used what is called a DQ-BSA assay. So basically this is bovine serum albumin that has been heavily dotted with fluorescent fluorophores. And so heavily that actually they're so near each other, they actually quench their fluorescence. So although they're carrying fluorophores, the whole protein basically is not fluorescent. However, if the protein is being cut, that means if it's being proteolytically degraded, you get single fluorophores with no nearest neighbors, and then they are able to fluoresce on their own. So basically that means the more degradation of this compound you have, the more fluorescence you get. And this is actually a very nice tool to look at protein degradation and to see whether an organelle is actually degradative. So here again, you have macrophages. Here you have the DNA stained of the nucleus. Here is the DNA of an internalized Borrelia. And you see that this is very much positive for this dequenched BSA signal. So this means the Borrelia, the compacted Borrelia, is actually present in a compartment that is proteolytic. And if you then look at what happens if you delete one of these effector proteins, mainly RAP5A or RAP22A, by 2 different siRNAs, that means you get a decrease of this fluorescent signal. So that means if you decrease one of these critical proteins, you also get less degradation of the Borrelia inside the cells. So the activity of both GDPases is actually absolutely required to get very good degradation of the Borrelia inside of the macrophages. So what's the consequence then? So, and Xenia asked then, is it possible that then the Borrelia would be able to actually survive intracellularly? Admittedly, that's an artificial system, but still. And what she set up is what we call an intracellular survival assay. So basically, we have the macrophages sitting in the petri dish. We infect them with Borrelia, let them being internalized, destroy the extracellular Borrelia by antibiotics. And then at a time point where we don't have full degradation, we disrupt the cells mechanically. And then we try to reculture previously internalized Borrelia that might still be viable. And this is actually what happens when you do this experiment in a control cell. So this is actually fine-tuned to conditions where you actually are able to reculture at least some of those Borrelia. So you have really to fine-tune that, that you don't get a total loss of intracellular Borrelia. However, you have way more surviving Borrelia if you disrupt cells that have been depleted for RAP22A or RAP5A. So that means, again, these two RAP GDPases are not only necessary for the lytic machinery to be recruited to the Borrelia phagosomes, but this actually also results in better survival of the Borrelia, of the spirochetes, if you actually deplete these GDPases. And I have to add, one of the nice angles from this line of research was that came from the idea how actually are these RAP5A vesicles getting in contact with the RAP22A positive phagosomes. And this idea has been floating around in the literature that it actually might be the endoplasmic reticulum that is present throughout the whole cell, that this is actually one of those organelles that might be used for getting into contact with these phagosomes. So the movie you're seeing now is RAP22A, here positive at the Borrelia phagosome, and this time in magenta. This time in white, you see the endoplasmic reticulum with these tubules, and again in green, the Borrelia. And what you see is really that during the uptake of the Borrelia into the macrophage, this endoplasmic reticulum really spins a web around the phagosome that really enwraps from the cell internal side the phagosome and really actually draws it into the more internal parts of the macrophage. You see that way better here in the gallery. Again, this would here be the outline of the surface of the macrophage. This is the internalized Borrelia present in the phagosome. And you see here the spider web of the endoplasmic reticulum that actually intricately enwraps actually the phagosome. And this might actually be the way how these RAP5 positive vesicles are able to contact the phagosome, because we've been, I always tell my students you should read more literature, and we all should. And actually by looking at the literature, we found that vesicles or RAP5 vesicles actually are in constant contact with the endoplasmic reticulum. So this is work from the lab of Gia Feltz. And we actually look at our macrophages, whether this is true also in them, and it actually is. So this is, again, staining for RAP5 positive vesicles. And as you can see here, they are really part of this web where they are always in close contact with the endoplasmic reticulum. And if you imagine then that the endoplasmic reticulum actually enwraps the RAP22A positive phagosomes, you can easily see that the RAP5 vesicles are actually being brought along as the ER enwraps the phagosome. So the first model that I'm presenting here for the internalization of Borrelia is, I've shown you first that there's an uptake structure, the coiling pseudopod, and if this is contracting, you actually have at the same time, because the Borrelia is so elongated, already the internalization taking part. And this happens by RAP22A being recruited to the phagosomal coat, and the ER, the endoplasmic reticulum, enwrapping that nascent phagosomal coat. And since these RAP5 positive vesicles are present at the ER, they're being dragged along and they are actually brought to close vicinity to the phagosomal coat. And at sites where these RAP5 positive vesicles contact the phagosome, you get the formation of these membrane tubules that lead to the shrinkage of the surface of the phagosome and the eventual compaction. And compaction is a necessary prerequisite for actual degradation of the Borrelia. And this actually means if the Borrelia are able somehow to evade this whole machinery, they also might be able to evade the intracellular degradation. So I told you that the membrane tubulation at these membrane containing phagosomes is what actually captured our interest. And this nice guy, Matthias Klose, who was a graduate student in the lab and is now a postdoc in the lab, decided to look at potential regulators of these membrane tubulation events. And a family that is actually involved prominently in membrane tubulation are the so-called sorting nexins or SNXs. There are a couple of them. You can see it doesn't end with sorting nexin 31. But all of these sorting nexins have in common a so-called PX domain that binds to phospholipids that are present in membranous compartments. And actually our initial assumption was that one of these sorting nexins that would contain a bar domain would be one of those that is responsible for the membrane tubulation, because this bar domain is actually able to sense and also to induce membrane curvature events that are necessary to tubulate membranes. Never make such an assumption, because although we found one of those that is involved in membrane tubulation and also has a bar domain, SNX1, the most prominent SNX to localize at Borrelia phagosomes is also the smallest member of the SNX family, that's SNX3, which basically more or less only consists of a PX domain binding to phospholipids. So this really caught our attention, and Matthias decided to really look at SNX3. So this is a construct of SNX3 in magenta, GFP-fused SNX3. You see that it nicely localizes to the Borrelia that is being internalized, that is already internalized in the cell. And you also see, since there's a very good antibody for SNX3, that also the endogenous form really nicely localizes here to the internalized Borrelia. And if you look at that in a higher magnification and a 3D animation, you see that the Borrelia here in green is really again enwrapped by this SNX3 signal. So SNX3 prominently localizes to Borrelia phagosomes. What's the relation actually to these vesicles and to the phagosomes that I've been talking about? And what Matthias did here is he looked at internalized Borrelia in green, he looked at SNX3, and he looked at RAP5A. And you already see here in this yellowish color, these RAP5A positive vesicles. And what you could actually already see is that some of these RAP5A positive vesicles are also positive for SNX3. And you see that this is a very dynamic co-localization. And actually, this SNX3 positive phagosome gets repeatedly contacted by vesicles that are positive for both SNX3 and also for RAP5. You see that way better here. This is one cell, this is the internal part of one cell, and you have several uptake events. You see here this compaction state, that phagosome gets positive for RAP5. Here the next comes along, it gets positive for SNX3 in magenta. Here's the next one, again positive for SNX3. So that means this sorting nexin is really a prominent component, both of the phagosomal coat and also of these RAP5 positive vesicles. And that led us to the hypothesis that actually these RAP5 positive vesicles might bring in the SNX3 that is then being handed over to the phagosomal coat. Is SNX3, before we started on this line of research, it was of course important to ascertain that SNX3 actually has a role in phagosomal compaction. And it does, sorry, because if you really knock down SNX3, you see that you have a large decrease by about 60% of phagosomal compaction. And interestingly, the one that we suspected would be the most important one, the SNCS1 that contains this membrane curvature inducing bar domain, was not effective at all, not singly and not in combination with SNCS3. So SNCS3 is actually the sorting nexin that is most important, at least from the ones that we tested for Borrelia compaction. And again, you already know now this DQ, this dequenched BSA assay. We looked at the phagosomes that are positive for Borrelia DNA, that are also positive for a fluorescent dequenched, that means proteolytic compartment. And you see if you knock down SNCS3 by 2 individual siRNAs, you get specifically in these Borrelia containing phagosomes, but not overall in the cell. In the Borrelia containing phagosomes, you get a reduction by about 50 to 60% of proteolytic capacity. So that means SNCS3 is really important also to degrade intracellularly the Borrelia. And following that, if you do this intracellular survival assay in cells that have been depleted for SNCS3, also the cells that are depleted are actually a better or a more lenient host for the Borrelia that are able to survive to a higher degree in these cells. So how actually does that work? Because that's been puzzling to us, because the SNCS3 actually just contains more or less the PX domain. So what does the PX domain actually bind to? And then we really looked at phospholipids at these Borrelia containing phagosomes. And here you see the Borrelia phagosomes and various phospholipids, phosphoinositol 3P, phosphoinositol 3,4P2, and phosphoinositol 4. And all of these are somehow to a certain degree enriched at these Borrelia containing phagosomes. And what we really looked at then biochemically, and what Matthias could really show, what has been known, is that SNCS3 actually is able to bind PI3 and also to bind PI4P. And he finds that biochemically on membranes that have been spotted with different phosphatidyl inositols, if you overlay these with an expressed construct of the SNCS3 PX domain, only PI3P gives you a good signal. So this PX domain is only able to bind PI3P to a certain degree. And this is exactly what we find here at these Borrelia containing phagosomes. And most importantly is that the PI3P fluorescence intensity always coincides with the most fluorescence intensity for the SNCS3 that you get here. Is PI3P really important for the Borrelia compaction? Yes, it is, because if we add an inhibitor that inhibits the kinase that is responsible for producing this phospholipid, you get a total decrease of the SNCS3 signal at the Borrelia containing phagosomes, and you also get way less compacted Borrelia in these cells. So binding of SNCS3 to the PI3P in the phagosomal coat is important to recruit SNCS3 and also for the subsequent compaction of these phagosomes. And he could really nicely show that by depleting cells of SNCS3 internally, you get a reduction by about 50% in compaction. If you re-express the SNCS3 by an siRNA insensitive construct that is not hit by these siRNAs, you get a full reconstitution to normal levels of compaction. However, you can't do that by an siRNA insensitive construct that has a point mutation that is no longer able to bind the PI3P. So SNCS3 and its binding of the phospholipid PI3P in the phagosomal coat is important, at least for a certain part of these compaction events that lead to degradation of Borrelia. So I told you SNCS3 is really tiny. Luckily for us, there has been a crystal being published of SNCS3, and SNCS3 here you see here in these crystallography depiction here in blue. And it has been known for quite a while that SNCS3 is able to bind the retromer complex, which is a complex that is involved in membrane tubulation, which got our interest. However, What you see here at this end is a C-terminal extension of the SNCS3 that has been totally uncharacterized. And the large barrel that you see here is actually the folded phospholipid binding PX domain. So these are these different domains of the SNCS3. And Matthias then decided to make really truncation mutants, as RNA insensitive truncation mutants that cover all the possible variations of the PX domain, the C-terminal extension, and the N-terminus being present here. And to cut a long story short, what he found is that the N-terminus is to a certain degree important, but not really totally significant. However, if you cut off this tiny C-terminal region, which is actually just 11 amino acids, the SNCS3 is no longer able to localize to the Borrelia-containing phagosomes. And this again, it looks complicated, but actually it's quite easy, although quite labor intensive, I have to say. Let's concentrate here on the right. This is the compaction assay that I've shown you before in cells that have been depleted by siRNA for SNCS3 and re-expressing siRNA in sensitive constructs. So here you see the control, you get about 50% decrease of compaction in siRNA SNCS3 depleted cells. You get a full reconstitution with this RNA insensitive full-length construct. If you delete the N-terminus that binds retromer, that is supposed to actually be involved in membrane tubulation events, this is not really the point here. However, as soon as you get rid of this tiny C-terminal portion, basically you get down to basic levels. So the C-terminus of SNCS3 is really important for both the localization at phagosomes and also the compaction of phagosomes. And what Matthias then did is he expressed solely the SNCS3 C-terminus and fished for binding partners in lysates of primary human macrophages. And this is a list of what came out of the mass spectrometry analysis. And what caught our attention is this protein called Galectin-9. And you can see here in these co-immunoprecipitation events that this is actually the elution. So in the elution, you get the SNCS3 that has been coupled to the GFP, actually just the C-terminus of the SNCS3. And here you see the Galectin-9 and a small portion of the intracellular part of the Galectin-9 binds to SNCS3. Now you could say that, well, this is maybe an overspill of this lane here in the gel, but it's actually not, because Matthias could very nicely show that if you look for the Borrelia signal, which is way more intense than the Galectin-9, from the exact same blot, you just get that in the flow through, not in the elution. So what we really think is what happens here, the SNCS3 C-terminus at a certain time point is able to bind a subset of the intracellular Galectin-9. Well, that's biochemistry. And you might have surmised from what you've seen before that I'm a guy who likes to look at things. And this is actually how it looks like. Does Galectin-9 really localize to SNCS3 positive phagosomes? And the answer, of course, is yes. The Galectin-9 here is in yellow. The SNCS3 here is in magenta, and here the Borrelia is in green. And you have one macrophage with several uptake events of Borrelia, and all of these uptake events here in the single cell show that the phagosome for Borrelia is positive for Galectin-9. And at the same time, if you deplete Galectin-9 from these cells, you again get a reduction of 50% of compaction of these phagosomes. The same value you get for SNCS3, and actually if you deplete again SNCS3 together with Galectin-9, you have no additive effect, showing that both of these proteins work in the same pathway. And this is what it looks like dynamically. The Borrelia turns up in green, and the Galectin-9 here is actually present in the form of these vesicles, and you see that at a very later stage, the Galectin-9 vesicles really are recruited to the Borrelia containing phagosomes, and then they actually also enwrap these phagosomes. this is just for the specialists. Galectin-9 is actually a carbohydrate binding protein, and therefore it has two carbohydrate binding domains, CRD1, CRD2. Matthias deleted all of these, again, just like the assay with the SNCS3, re-expressed as RNA-insensitive constructs, and he could show that both of these CRD1 and CRD2 domains are important for the compaction process. However, if you insert specific point mutants that are known to bind specific carbohydrates, you don't get really a clear answer. So the main message from this is that you need both of these carbohydrate binding domains, but none of the prominent binding carbohydrate binding partners of these domains seem to be involved in the compaction of Borrelia. So this is actually what a certain point of full stop in the lab. However, we have really now looked at independent marker proteins for these vesicles, and we found that a protein called flotillin-2 is also very much enriched at these galectin-9 vesicles, which gives us a second independent marker to really look at these vesicles and what happens if we knock down galectin-9. And this is an ongoing project in the lab. What I want to show you as a last line of research in this part is that is it then really true that SNCS3 is regulating RAB5A and galectin-9 recruitment to phagosomes? And you have here the galectin-9 again in these vesicles in yellow, the Borrelia in green, and the RAB5A here at these vesicles in red. And what Matthias could really nicely show is that if we look at the recruitment in this tiny volume around Borrelia containing phagosomes, if you look at that in cells that have been depleted for SNCS3, you get less RAB5A and you get less galectin-9 at these phagosomes. So the SNCS3 is really important for bringing both of these proteins to the surface of these phagosomes where they can exert their activity that leads to the compaction and eventual degradation. So that leaves us with a model of how SNCS3, despite being so tiny, is actually able to drive the compaction of the Borrelia. And this is here, you would have an internalized Borrelia present in, already inside a phagosome that is positive for these yellowish dots of RAB22. And you have this phospholipid PI3P present at the phagosomal coat. And I have to say this phospholipid is especially present at sites of high membrane curvature, because this is where the kinase that produces the phospholipid is especially active. That means due to the helical shape of the Borrelia, you get these phospholipid-rich domains, especially at the twists and turns of the Borrelia phagosome. And this is actually where the phospholipid attracts these vesicles that are positive for both RAB5A and SNCS3. And by binding of SNCS3 to these PI3P-rich domains, the vesicles that are positive for RAB5 are being recruited very closely to the phagosomal coat. And this is how actually RAB5A is brought into close contact with these RAB22A positive phagosomes. And at a later step, this tiny 11 amino acid extension of SNCS3 is able to recruit a second separate set of vesicles that is positive for galectin-9, that is also important for the downstream activity of compaction downstream of SNCS3. However, we still have to be able to figure out how galectin-9 works in this process. So to sum that up, what I've shown you is that the uptake of Borrelia by primary human macrophages is actually a two-step process. First, you have phallopodia that immobilize the highly motile Borrelia at the surface. This gives the coiling pseudopod time to closely enwrap the spirochete at the surface and draw it into the macrophage cell body. Once the Borrelia is internalized, this phagosomal coat gets enriched with RAB22A and is being contacted by RAB5A positive vesicles that are also positive for SNCS3. I showed you earlier how that works. SNCS3 is the adapter that brings in these vesicles, binds to the phospholipid this leads to the close contact with RAB5A and also recruitment to these galactin-9 vesicles. And all of this machinery drives the extrusion of these membrane tubules that lead to a shrinkage of the surface of the phagosome. And this is a prerequisite actually for subsequent degradation in these cells. And just as a very last step, and this is actually unpublished and a bit more eye candy, what we've resorted to is something that is not limited to the diffraction limit shown in immunofluorescence, but what we looked at is now the interaction of the primary human macrophage with Borrelia by a technique called FIB-SEM, which is actually a combination of electron microscopy tomography with the ablation of small portions of a cell by focused ion beams. So focused ion beam scanning, electron microscopy tomography. Sounds complicated, I'm going to guide you through that. What we actually did is we let primary human macrophages eat Borrelia that express GFP. This leads to the GFP signal here. This is the nucleus of a primary human macrophage. And then we embed them in ultra-thin resin that leads to fixation of the specimen. And this then can be accessed by so-called FIB milling. So that means you have a laser that really slices tiny portions out of this resin embedded specimen. And this then leads here to these slices that you can look at from the side. So here you would have the macrophage, here you would have the resin, here you would have the glass slide where the macrophage sits on. This is the nucleus, and this is actually what you've been seeing before. Also in other talks, the internalized Borrelia with its twists and turns. And if you then put one of these slices, one after the other, and animate that, you can actually go horizontally sectioning the macrophage with internalized Borrelia. So here you would have the macrophage, here are the Borrelia in contact with the surface, and here are the rising coiling pseudopods. And then you track slice by slice the extension of the Borrelia that have been internalized by the macrophage and falsely pseudocolor them. And then you also pseudocolor the membranes of the host cell, here in yellowish. Well, I say that so nonchalantly, but actually that's the work of hours and hours. But in the end, you get this pseudo-colored image of the macrophage surface here in yellow, together with these turquoise colored Borreliae that are in contact and embedded in this surface extension of the macrophage cell. And what you can already see here, you have huge convolutions of the surface that are in contact with the cell, the Borrelia cell, and also enwrap the cell. And what really captured our attention is not only these protrusions, but also the internalized membranes that you see here. And normally you would expect that you have a phagosomal membrane that closely follows the outline of an internalized Borrelia. And to our surprise, repeatedly we find these membranes of phagosomes that extend way deeper into the cytoplasm of the primary human macrophages than the actual extension of the Borrelia is. This again is the same cell, and here you have these membranes pseudo-colored in blue. And I've talked previously about the endoplasmic reticulum, and the endoplasmic reticulum is here colored in magenta. And this is animated in space. And you see that the Borrelia is actually internalized in these membrane folds, but the membrane extends way deeper into the cytoplasm than the actual Borrelia is. And this is actually now what this is then a more extensive rendering of the endoplasmic reticulum, which actually reflects what you've seen in the movies, that you have really this intricate web of the ER enwrapping the internalized Borrelia. And this structure that extends way more into the cytoplasm than the Borrelia, we now refer to as a tunnel. And we think the formation of these tunnels might actually be due to the partial extrication of the Borrelia from the host cell. And now I come back to these periplasmic flagella that have been here colored in this light bluish color that propel the Borrelia and lead to this rapid movement. And you have seen in the initial movies how the Borrelia struggle not to being taken up anymore. And we think actually it's not that the Borrelia, once captured, are being passively dragged along into the interior of the cell, but actually that they resist and they try to extricate themselves from the cell. And these extrication events might actually then lead to the formation of these tunnels where previously the Borrelia have been present here and then partially got out, but still got captured at the surface and then would be internalized eventually. So we tried to look at this process in a bit more detail, and what you see here is a phagocytic cup. So this is the surface of a cell. It could be a primary human macrophage. This would be a beet, but it could also be a Borrelia, and this is a phagosome. And what you can see here is that the outer surface of the plasma membrane is now the inner surface of the phagosome and vice versa. And the interesting thing is that it has been shown earlier by the group of Sergio Grinstein that one of the phospholipids that's present only at the inner surface of a plasma membrane, phosphatidylserine, is then present at the outer surface of phagosomes. There's actually a reporter construct from the protein called lactaterin, and the domain, the C2 domain of lactaterin actually binds to phosphatidylserine. So we really looked at, can we recapitulate in normal immunofluorescence what we see in FIB-SEM? And this again, here is the surface of a primary human macrophage with a partially internalized Borrelia cell. And here you indeed see that the LACT-C2 staining, which indicates phosphatidylserine at the phagosomal outer membrane, enwraps the Borrelia. And it enwraps, extends more deeply into the cytoplasm than the actual Borrelia is. So you really can observe these partial extrication events, because here again, this is an inside-outside staining. This is an antibody staining showing that the Borrelia cell is still partially outside, but the antibody can no longer go into these nascent phagosomes. So, and we think that in these partial internalization events, the Borrelia, where still let's say a considerable part is being outside and still can be propelled by the periplasmic flagella, then the Borrelia might still be able to extricate itself a bit and then form these membrane tunnels that you see here. So that leads us to the final model of this presentation. I've shown you that Borrelia are being immobilized and enwrapped at the macrophage surface by phallopodia and coiling pseudopods, that they are compacted and driven to degradation by these membrane tubulation events and phagosomes. And what we think happens in between at a certain portion of partial uptake events is that although they are being partially internalized in phagosomes, once still a part of the Borrelia is outside, it might be able to lead to a propulsion event of the Borrelia, and the Borrelia might try to extricate themselves partially or fully and lead then to the formation of these Borrelia devoid membrane tunnels that still retain their contact to the ER that you usually see only at these fully formed phagosomes. And with this, I want to close. Thank all the busy and highly motivated lab members, all our collaborators, and especially you for listening. Thank you very much.